So yeah, it's really my pleasure to be here today. Thank you all for showing up to my defense, both in person and on the Zoom. Thank you guys for the really wonderful intros. I'm just going to dive right into the talk portion so see how much we get through. And then hopefully we'll have a moment at the end for some questions. And Stephen, you can let me know if anything's going wrong on the Zoom. But for now, I'm going to assume everything's good to go. So the title of my talk today is Protein Biomarkers of Human Organ Aging and Neurodegeneration. And let's see if we can get the slides moving. There we go. Right. And I'm going to maybe occasionally gesticulate with the mouse just so people both in the room and on Zoom can see. So just to orient everybody on the talk outline, we'll go through a little bit of background on aging and neurodegenerative disease and on proteomics. And then I'm going to try to tell you guys two stories today. First, a little bit of work on Parkinson's disease, and then some work on aging more broadly using proteomics. So let's just dive right in. So the focus again today is aging. And aging is really a key driver of disease and many different categories of disease. So it's the number one risk factor for Alzheimer's disease, heart disease, cancer, and many other kinds. And really one of the major goals, I think, of modern biology is to understand what it is about the aging process that predisposes us to these many different categories of disease that we wouldn't otherwise think of as sharing core mechanisms or hallmarks. In order to get there, we really need more interpretable and quantitative biomarkers of the aging process itself for us to understand what's happening at the molecular level that leads to all of these different outcomes later in life. So just to zoom in on one disease, which is going to be part of the focus of this talk, if we take Alzheimer's disease, which is the most common neurodegenerative disease, aging really dwarfs all other risk factors for the disease. So if we look at all risk factors that we know of for Alzheimer's disease, genetic, environmental, and otherwise, and compare it to the risk conferred by 25 years of aging, the risk conferred by aging is really much larger. And this is such a large effect that if we could delay the effects of aging on the brain by just 5 years, we could prevent 50% of all Alzheimer's disease cases. And end-stage brain aging in general leads to neurodegenerative disease. So the two most common consequences of brain aging at the end of life are Alzheimer's disease and Parkinson's disease, which are the number one and number two most common neurodegenerative disorders. So Alzheimer's disease is characterized by progressive memory loss and cognitive decline, and PD, the hallmark symptom, is a progressive loss of motor control. So these are both really exponentially related to aging. So they affect about 13% of adults worldwide over the age of 60 and 35% of adults over 85. And at the time of death, if you look in people's brains, nearly all older adults, regardless of if they had symptoms or not, have some evidence of the underlying pathology for one or more of these diseases. So in the Alzheimer's field, we sometimes say that everyone is a pre-symptomatic Alzheimer's disease patient. It just is unsure if you will live long enough to experience the symptoms. And you can kind of see this on the brain scans on the left as well, where both in healthy and Alzheimer's disease brains, there is neurodegeneration that accumulates with aging. So the number of people affected by diseases is expected to triple by 2050 as more people are living long enough to experience the symptoms. And right now, there are very few treatment options for these conditions. There's no cures, and it's debatable whether there are any drugs that meaningfully slow disease, but in general, there aren't. These are also very difficult to detect early, and it's thought that both in AD and PD, there's decades of silent symptom or silent damage accumulation before we actually see symptoms. So we really need better biomarkers of brain aging to understand these diseases and the process overall. And we need it for many reasons. The first is just accurate diagnosis. So about 20% of all AD and PD cases are misdiagnosed, and rare forms of dementia are almost always misdiagnosed as AD or PD. And of the people who are misdiagnosed, about 70% of them have inappropriate disease management clinically, even with the very limited treatment options that we have available today. We also need it for better prognosis. So right now it's very difficult to tell how patients will progress in these diseases. There's no test to do this, and some patients decline quickly while others take many decades to do so. We really need this for early detection as well. So there's no tests to detect AD or PD before the characteristic symptoms of memory loss or motor loss occur. But we know from brain imaging studies, as well as a number of other methods, that these diseases actually start decades before this. And it's increasingly thought that by the time these characteristic symptoms appear, it might be too late for a complete cure because there's been so much damage that's accumulated in the brain. And all of this impacts our ability to do effective drug development and create clinical trials for these diseases because it's difficult to treat patients early enough. We can't really detect who is in need of treatment. And it's also difficult in the context of a clinical trial to identify which patients are actually going to decline fast enough to see a benefit within the time frame of the trial. So if you have a bunch of patients who are going to be stable for many decades, it is a very difficult thing for the trial and could, basically run past the clock. In terms of cost. So these are big impediments for drug development, which is a big impediment for future treatment. And it's also an impediment for basic research for many of the same reasons. It's hard for us to study the early disease process, and it's hard to study which factors in the disease might be causal at the molecular level because it's just hard for us to find the right patient populations to even study. So there has been a lot of work on AD and PD biomarkers in the last 50 years, but they've been largely dominated by a single strategy. So AD and PD both have obvious changes in the brain if you look at people's brains at autopsy. And these are particular proteins that aggregate in the brain in Alzheimer's and Parkinson's. So in Alzheimer's, there are two proteins that aggregate in the brain, tau and amyloid beta. And in Parkinson's disease, there's just one protein that aggregates called alpha-synuclein that forms an inclusion inside of neurons called Lewy bodies. And there's been a huge focus on these proteins in basically efforts to try to detect them early in the cerebral spinal fluid, detect them early in the blood, try to image them in the brain. And using these methods, we have made a lot of progress, especially in Alzheimer's disease for diagnosis, but not enough to solve any of the challenges that I mentioned on the previous slides. And it's not really clear that it'll be enough just pursuing these approaches in the near future either. So we need alternative strategies. And a different strategy that I spent most of my PhD trying to explore was to take a data-driven approach where we just try to quantify as many molecular components in the disease and aging process as possible, and then look for patterns that we might be able to use as biomarkers and to understand the early disease process. So this is generally sort of called omics when you're trying to quantify comprehensively all the molecular components of a system. And just to make it concrete with an example, we'll look at blood, which is an incredible window into aging, which I'll get to in the last third of the talk. So there are many tens of thousands of components in the soluble fraction of blood, which is called plasma. And we could try to comprehensively profile all of the proteins, for example, in this fraction. And this would be an example of proteomics. So there's multiple methods to do this kind of measurement, and I won't really get into that, although I'm happy to discuss specific methods in the Q&A if people are interested. But this is sort of an example of what that kind of data looks like. So this is data coming from Tony's lab, where we measured about 3,000 proteins in a population of a few thousand healthy people spanning the adult lifespan, and plotted in this heat map on the rows. Each row is a protein, and each column is the average level or expression level, or just number of protein molecules in a specific fraction of plasma in the population. So the columns are averages, rows are proteins. And then the colors here is a z-score, which is a normalized measure of concentration. And you'll see that term z-score, which is sort of just a normalized measure of effect size throughout the presentation. So we can see based on this plot is when we do this sort of proteomics, one of the things that stands out is that most proteins change with age. And this is pretty exciting because it means there's lots of signal for us to explore in this kind of data, but it's also challenging because it's difficult to make sense of these large and complex omics data sets. So today I'm hoping to tell you two stories where we try to make sense of this kind of omics data, and I hope it can convince you by the end of the power of this kind of approach for developing biomarkers for aging and for neurodegeneration. So the first story is titled Discovery of a Novel Fluid Biomarker for Parkinson's Disease with Multi-Tissue Proteomics. And I already told you a little bit about PD, but the other major thing you need to know about Parkinson's disease is that dopamine is very important in Parkinson's disease, and many of the symptoms are primarily caused by a lack of dopamine in the brain. So neurodegeneration in Parkinson's disease happens in a very specific brain area. that's called the substantia nigra, pars compacta. It's in the midbrain. And this is a place where dopaminergic neurons in the brain reside, and it's sort of the production site for most of the dopamine in your brain. And in Parkinson's disease, these neurons degenerate, and this causes the sort of characteristic symptoms, which include problems with motor control, as well as automatic functions like bowel movements and problems with mood. So the standard of care for Parkinson's disease currently is L-DOPA therapy, which is essentially just trying to supplement dopamine into the brains of people with Parkinson's disease. So they stop producing it because the neurons die, so we try to give it to them exogenously. DOPA is a precursor to dopamine that your body transforms into dopamine after it crosses the blood-brain barrier. So as I mentioned already, there are really very few specific biomarkers for Parkinson's disease at any stage. And PD actually starts long before motor symptoms appear. It has this long pre-symptomatic stage that's called a prodromal phase. And we have no markers for this phase at all. But we also have no markers even for accurate diagnosis at the time symptoms arise. So right now, diagnosis is done entirely based on clinical presentation. And we don't have any markers looking into the future to tell people about their prognosis, to inform clinical management, drug development, figure out how fast people are declining, or if medications they're taking are helping. And along this entire disease course, we don't have any confident molecular correlates of the underlying disease process really to speak of that are quantitative. So in order to try to address some of these questions, I went into a study that has been conducted here at Stanford in a multi-omics disease cohort called the Stanford Alzheimer's Disease Research Center cohort, which is a group of about 700 people enrolled across 5 studies here who span disease conditions, Alzheimer's disease, Parkinson's disease, as well as healthy aging studies. These people have cognitive testing, health assessments yearly. We have some brain MRI, as well as a number of different omics technologies. And most importantly for this talk, we have plasma proteomics from blood and cerebrospinal fluid proteomics from the spinal cord. And so to get into the study a little bit more, we use a proteomics platform called Olink to quantify about 1,200 proteins in these patients. We have about 250 samples, about 170 of them are matched. And again, these people are spanning healthy Alzheimer's and Parkinson's disease in cerebrospinal fluid and in plasma. For those in the audience who are more familiar with omics, Olink is a kind of proteomics platform that's a multiplexed proximity extension assay. So it's essentially like a super multiplexed ELISA with a sequencing readout. And we use this to basically quantitatively measure the protein concentrations in blood and cerebrospinal fluid of these patients. And when we look at the concentrations of these 1,200 proteins in Parkinson's disease, one really sticks out in particular. So this is a plot where we're looking at protein concentrations of people with Parkinson's disease versus healthy controls. So the way you read this plot is on the x-axis is a measure of how strongly a protein is up or down in the disease. And on the y-axis is a measure of the statistical significance of that finding. And you can see in this plot that there's one protein that really sticks out as being up in Parkinson's disease called DDC in cerebrospinal fluid. And this also really sticks out in the plasma as well. And when we compare Parkinson's disease to Alzheimer's disease, we see the same finding. So this is a finding that seems to be specific to Parkinson's disease rather than to neurodegeneration as a whole. And we thought this was immediately an intriguing finding because we already know this protein is pretty important in the underlying disease process of Parkinson's disease. So DDC codes for an enzyme called L-aromatic amino acid decarboxylase, which is actually an essential step in the dopamine synthesis process. So it catalyzes the last step of dopamine synthesis from DOPA to dopamine in the brain. And in the brain, it's specifically expressed by aminergic neurons in the substantia nigra, which we can see on this body-wide plot of RNA expression levels. And one thing that we saw in our cohort is that AADC or DDC is highly upregulated in Parkinson's disease compared to both healthy controls and Alzheimer's disease subjects. And this is true even for Parkinson's disease patients who we define as being very, very early in the disease, which is within a year of diagnosis in this case. So we wanted to explore this a little bit further. And one of the first things we looked at was to see if we could see a quantitative relationship between these levels of this protein and some of the symptoms of the disease, which is something that we don't have for Parkinson's disease biomarkers in general. So on this slide, I'm showing a measure of that. So we're looking at the levels of AADC compared to 2 measures of motor symptom severity that are performed by a clinical test called MDS-UPDRS-3, which is a test that's delivered by a neurologist in a clinical appointment. And what you can see is that there is a general correlation that's highly statistically significant between ADC levels and motor symptoms in our cohort. So we thought this was a really interesting finding to start, but there is one major caveat to this protein as a potential biomarker, which is this question of L-DOPA treatment. Since this protein is directly involved in dopamine synthesis, it's feasible that some of this effect could be explained by the fact that people are taking a medication to try to increase dopamine synthesis in the body. So in order to really rule this out or understand the impact of dopamine therapy in Parkinson's disease, we really need a treatment naive cohort of people who aren't on the therapy to disentangle this effect. And this is actually a really challenging thing to do in Parkinson's disease because pretty much everyone takes this medication and they start taking it very early. It's really the frontline therapy and there's not much else that we can do for these patients. But luckily we were able to find a cohort where we have people who have yet to start the therapy. And this is coming from the Parkinson's Progression Marker Initiative, which is an international study of de novo Parkinson's subjects, which means that they're diagnosed at the time they enter the study, so they haven't been on any medications. And it also has a study of high-risk individuals with genetic risk factors for Parkinson's disease. And we have many years of clinical follow-up in motor symptom testing on these people. And we were able to get access to two exciting data sets in this cohort. One is a replication of Olink in plasma proteomics using the same proteomics platform, where we have about 70 people, and they have sampling over 4 years. Excuse me. We also were able to get an orthogonal proteomics platform, mass spectrometry, looking in urine of people from this cohort, where we have a much larger sample size and includes some of these people who have genetic risk factors who are in a prodromal phase. So we think they'll develop Parkinson's disease, but they don't have symptoms yet. And so first looking at the replication cohort in the Olink CSF and plasma proteomics, we see that again, in these people who are very early in the disease, like this is right on diagnosis where they're getting this sample, and they have not taken dopamine replacement therapy, DDC is still the top upregulated protein in these people. And this is what that looks like on a per sample basis. And we see the same general trend that there's a statistical relationship between AADC levels in the cerebral spinal fluid and measures of motor symptom severity in this cohort. And this, I think, is actually a pretty strong evidence because these people really don't have very many motor symptoms. So the dynamic range of this test is pretty small at this point. So the fact that we see a relationship here is pretty encouraging. We also see this relationship in the plasma, and it's even a little bit stronger. So we see the same relationship with motor symptom severity, and we also see a relationship with cognitive symptoms. So on a test of cognition, plasma levels of ADC are also negatively associated with cognition. So higher levels of ADC in the plasma sort of relate to lower cognitive performance, which is an interesting finding for the disease. So we also wanted to test if we could use the levels of this protein in blood and cerebral spinal fluid to try to diagnose Parkinson's disease with a molecular test. So we trained a machine learning model to classify people as Alzheimer's disease, Parkinson's disease, or healthy control based on the levels of DDC, either in plasma in yellow, or sorry, in plasma in red or CSF in blue. There's no yellow on this. So that's what this kind of plot shows. This is called a receiver operating curve plot. And on the x-axis is the specificity of the diagnostic test. So that means how many false positives were there, or to say another way, how many people without Parkinson's disease did we accidentally diagnose as having Parkinson's? And on the y-axis is the sensitivity, which is like how many people with Parkinson's disease did we miss with the diagnosis? So a perfect fit would be a right triangle on this plot where we have perfect sensitivity and specificity. And the diagonal line is a random guess. So we want to do better than diagonal for sure. And you do with this protein marker. So as you can see, the models do much better than that. And they actually have a pretty high area under the curve of around 0.8 in these two independent cohorts where we tested the levels. And so this is a really encouraging sign for the future application of this in diagnosis. It's not currently at the level where we could just go into the clinic and start using it like exclusively to diagnose people, but it's a really interesting finding that indicates we should really pursue this further. And finally, we were also able to look in mass spec data in the urine, as I mentioned, of people who have genetic risk factors for Parkinson's disease, but who don't have symptoms yet. So these are two populations of people who have mutations in these genes LRRK2 and GBA. And not all of them will go on to develop Parkinson's disease, but most of them will, about 80%. And in these prodromal cohorts, who again don't have any symptoms yet, we see that the DDC levels are actually highly elevated compared to healthy controls. And this is particularly true in the LRRK2 patients, which we think is an exciting finding because LRRK2 patients tend to lack some of the alpha-synuclein-based pathology. That's sort of like the other promising strategy for trying to detect and diagnose these patients. So on current other approaches, we often have a really hard time with these LRRK2 patients. And it seems to be a place where DDC actually does a really good job. So in summary for this story, just hope that we were able to show you that we think ADC is a promising potential biomarker for Parkinson's disease. It's one of the first molecular markers of any kind which correlate with symptom severity in PD, even at a very early stage. It has a very compelling underlying connection to the disease process and its importance in dopamine synthesis. And we think it could have utility in diagnosis, prognosis, and perhaps in early detection as well. It's gonna wet the throat for a second. I need to work on my speaking voice. All right, so on to the next part. The next part of my talk is titled Organ-Specific Aging Signatures in the Plasma Proteome Track Health and Disease. Thanks to everybody from the labs who's showing up yet again to hear me talk about this yet again. You all deserve a round of applause just for that resilience. Actually exciting thing here, this is work that was done with Hamilton O, who's another PhD student in Tony's lab. And this work was just officially accepted in Nature earlier this week. So, so for this, we're zooming back out to aging more broadly. And again, I just want to reorient us to this question of why does the aging process seem to predispose us to many different categories of disease at the same time? And so within this question and the need for more biomarkers to address this question, one of the things that comes up is how do we actually go about trying to measure aging comprehensively? And this is actually pretty challenging at the molecular level because we don't really know what aging is. We know some of the components of it, but we definitely don't have the full story, and we don't really know what's causal. So because of this, data-driven approaches that use omics are actually really valuable at this stage in the research in aging. And there's a pretty easy intuition for how we might try to use this kind of omics data to understand aging comprehensively. So if we go back to this plot, that's showing, again, average protein expression over the lifespan in a population of people across about 3,000 proteins. And we just take a cross section. We could try to look at this pattern across all 3,000 proteins and say people who are about this age tend to have this pattern in their proteomics data. And so we could kind of guesstimate this and say this pattern, if we see it, it looks like about 28. And we could also try to do this in a more rigorous way by applying statistical pattern recognition to this data to try and get a better estimate of people's ages. And this is essentially what we're doing in this next study. And this statistical pattern recognition has another fancy name now called machine learning. But that's really all we're doing here is trying to estimate the age of someone based on this profile by aggregating this score. And so this is the concept that's called an aging clock. And it's essentially just a machine learning model that's trying to estimate an age or something else that's close to an age based on a molecular profile. And so merging omics and machine learning in this way has become one of the primary methods to understand how we age at the molecular level. And this work has really been pursued across a number of different kind of omics types, but it was first pioneered within a different type of omics called CpG methylation, which is a kind of DNA modification. And the really interesting thing about these plots, other than the fact that you actually can predict someone's age from these profiles, which is in itself a bit surprising. And I guess just to walk you through the plot a little bit here, So on the x-axis is, we have a population of people, and on the x-axis is their chronological age in years. And then on the y-axis is the age that we're estimating based on their profile, just like I walked through in the last slide. And what you can see is we actually do a really good job of estimating people's ages based on a profile like this. And this tends to be generally true across many different kinds of omics, from DNA methylation to proteomics to RNA. But one of the most interesting features of these kinds of plots is that when we look at people who maybe have a different estimate than we would expect, so people who have a molecular profile that looks older than their age, they have a positive age gap, or a molecular profile that looks younger than their age, or negative age gap, these people are at actually a higher risk of death in the future, or a low risk of death in the future. And this is an extremely reproducible finding that we've seen over the last decade. And so this suggests that these kinds of models can actually, in an unbiased way, identify some of the molecular signals that are important in the physiological aging process, which motivates why people are studying them. So one of the key challenges, though, to these approaches that I'm talking about, these aging clocks approaches, is that they don't fully capture the complexity of human aging, because it's difficult to condense all of human aging to a single number. And that's because we know that there are many different outcomes to aging. Some people develop lung disease, some people develop brain disease, others kidney, others heart. And this aging heterogeneity is something that is difficult to capture with the current models in a really expressive way. And so one of the major goals of the field is to try to understand this aging heterogeneity between individuals and at the population level. And so that is sort of the motivation for our study here, is this question of, can we actually go to the level of individual organs and identify the aging process and track the trajectory of aging? And so to try to address this question, we are coming back to the blood and taking advantage of some really unique features of blood plasma, which is that blood is really the communication highway of the body. And because of that, it contains proteins from all tissues. And this is already something that's exploited in the clinic to try to measure organ function. So we have specific proteins that doctors will try to measure your liver function, your kidney function, your heart function, et cetera, that are circulating proteins that are derived from those tissues. So we already know that this is possible, that you can measure specific physiology of tissues using plasma. And we also know from prior work that you can build these very high-performing aging clocks from plasma proteomics, from work that was done in Tony's lab, as well as labs at the NIH before us. So combining these two insights actually leads to a very straightforward idea for how we might be able to measure organ aging at the population level using plasma proteomics. The first is to figure out, in as comprehensive of a way possible, which proteins in plasma are actually coming from specific organs. And then we can use that information to train organ-specific aging clock models. And then hopefully with that information and the information we already know about the biology of proteins, we can learn some new things about organ aging biology. So this is what we've done in this study. And in order to do this, the first step is to try to map the organ specificity of different proteins that are circulating in plasma. And to do this, we're using an RNA expression resource called GTEx, which has done deep sequencing of the RNA from every organ, not quite every organ, but many, many organs in the body of a large number of people. And so we define the tissue specificity of different genes based on their RNA expression levels. So genes that are only expressed in a single tissue, we call putatively organ specific. And then we can use this information genome-wide from GTEx to annotate a plasma proteomics assay where we're measuring about 5,000 proteins in blood. And this assay that we're mostly using in this study is the SomaScan assay, which is a different kind of proteomics platform from the one I talked about in the last assay, or in the last story. But the principles are largely similar, and I'm happy to talk more about the details in the question and answer period. Sorry, that was my phone. So like I said, we use this to annotate the plasma proteome. And when we do this, we see there are a number of proteins that we can detect on this assay that are tissue specific to a number of different organs, the largest number of proteins coming specifically from the brain. And so we can use these lists as input to train machine learning models to develop tissue-specific aging clocks, which is basically the setup for the rest of the study. So how we're doing this is we have multiple different human cohorts where we've measured plasma proteomics and we have health data over many years. And so we're training our aging clocks in a healthy cohort of about 1,400 people from the NIGHT ADRC. And then we can evaluate the performance of these models against a number of different health outcomes in independent cohorts that we also have proteomics in. And these are 5 cohorts listed here in about a few thousand people. So just a little bit more detail on machine learning and sort of the models that we're generating. So we are using what's called a bagged ensemble of lasso models. That's the machine learning approach, which I'm happy to talk about more in the Q&A if people are interested. But essentially, the output of these models we get is an aging clock that kind of looks the same as the plot that I walked you through in the intro. And we're training 3 different kinds of clocks here. We're training the organ-specific models that I just discussed, where we're using inputs from organ-specific protein sets. So those models only have proteins that are coming from the brain or from the heart or from the liver. We're also training a model that we call the organismal model, which is basically the inverse of those organ-specific models. So that's taking proteins that aren't specific to any organ. And then we also train a model that we call the conventional model, which is just able to use all proteins in the plasma, so it doesn't pay attention to the organ specificity in any way. In the organismal model and the conventional model sort of serve as benchmarks and controls to help us understand what, if anything, we're gaining or losing by really zooming into specific organs. And actually, before I go on, I just want to also reemphasize that the most interesting quantity that comes off of these models is this age gap, which is to say the deviation in someone's profile, the molecular profile from their chronological age. So that's really the quantity that we're going to be measuring throughout the rest of the study. And so to not bury the lead on this, this approach of generating organ-specific clocks leads to very high-performance models with some interesting properties. So one of the first ways that we decided to look at this was to look at future all-cause mortality. So again, looking at people's future risk of death based on their aging profiles. And 10 out of the 11 organ clocks that we trained are able to predict future risk of death in this independent cohort over a 15-year follow-up. So the way you read this plot is on the x-axis is a measure of risk. So a hazard ratio of 1 is a neutral risk, and a hazard ratio of 1.2 is an increased risk of 20%. And so people who have a 1 standard deviation increase in their heart age, they've accelerated heart aging of about 2 years, have a 50% increased risk of all-cause mortality over the subsequent 15 years. Yeah, and this is a pretty large effect, actually. It's on par with models that other groups have trained to specifically predict mortality. And we're not really specifically tuning the models for mortality. We're specifically just focusing on aging. So one other thing that we see that is interesting is that at the population level, organ age gaps aren't tightly correlated. So we see that there's largely distinct signals coming from these different organ models when we look at their residuals. So these age gaps aren't all traveling together, which is something you might expect naively. So this is summarized here in this correlation plot of the age gaps across different organs and also in this inset distribution. So you can see there is some correlation. So people with older brains do tend to have a little bit older livers and hearts and kidneys as well, but it's not like all the organs are traveling just really in concert together at the whole population level. So at the individual level, the relationships are actually a bit more complicated, but we think they're also very interesting. So these are just some examples of that. So at the level of different individuals, people with the same sort of conventional age gap can have a wide variety of different organ aging profiles, such as these 3 participants shown here. And we also observed that many people have kind of extreme organ aging in 1 or more of their organs, which we define as people who have more than 2 standard deviation difference in their organ age gap from the population average. And so to understand this a little bit better, we performed some unbiased clustering to look at different aging types in the population based on this extreme organ aging profile. And we found that about 20% of the population has an organ-specific aging type, which is really dominated by the aging of a single organ. And that's what's shown on this UMAP here, is an unbiased clustering algorithm to group people based on their organ aging profiles. And we see there are some people who have multiple aged organs, but it's a smaller portion of the population than I think we were expecting at first. And this is a sort of another way to visualize this here for 3 different clusters. So this is the average age gap in the kidney aging cluster, the heart aging cluster, and the multi-organ aging cluster. And sticking with these organ aging clusters that are defined by these extreme aging signatures, we do see that across a number of different diseases, there's a strong relationship between this accelerated organ aging and particular diseases. And some of them track pretty strongly with the accelerated organ aging itself. So for example, people in the heart aging cluster have a very strong a much higher likelihood of having atrial fibrillation or heart attack. People in the muscle aging cluster have a much higher likelihood of having an impaired gait based on sort of motor tests that are administered in the clinic. So looking at the whole population level, so now not really thinking just about these aging clusters, but just looking at the whole population distribution of organ age gaps, we see the same phenomenon across different organs. So regardless of age type, kidney aging shows a strong association with multiple diseases that are known to have a severe impact on kidney health, such as hypertension and diabetes. So people with hypertension have about a half standard deviation of extra accelerated kidney aging compared to the population average, which is about, again, 2 years of extra kidney aging. This is also true in the heart, where we see, compared to the other organs, the heart has a much stronger relationship with heart-specific disease traits that are age-related. So again, what we're looking at here is atrial fibrillation and heart attack, which are sort of two of the most common aging complications in the population. And we also see, again, now looking forward in time instead of looking cross-sectionally, if we look at people who have healthy hearts at baseline in an independent cohort, again, from the one that we trained in, so now, again, we're looking only at people who have clinically healthy hearts at baseline, meaning they wouldn't be diagnosed as having a high risk for heart disease. We can see over a 15-year follow-up period, particularly people with heart aging with accelerated heart aging are at a very high risk for future development of congestive heart failure. And this is about 250% increased risk. We also see some trends for other organs as well, but it's interesting that the heart in particular sticks out here. So kind of coming back to Alzheimer's disease, because that's just a major focus of the lab and a focus of our research. This is a phenotype that we've been very interested in characterizing with this approach. And it's been historically difficult to understand brain aging with these approaches. There hasn't been a lot of previous success with blood-based markers. Sometimes you can go into the brain itself and measure some of these omics profiles and see some strong relationships. But we really wanted to see if we could do this with our blood-based approach. And so our first generation of models did reasonably well, which I'm not really going to show the results for because we really wanted to get great performance when measuring brain aging. And so to do this, and this was really led by Hamilton, this additional round of model optimization, but we did an additional round of model optimization using a machine learning framework that we developed that's a little bit, I think, complex for this talk, but I'm happy to sort of walk through it again in the Q&A or offline with people. But we developed a feature permutation algorithm that essentially is able to measure the importance of different proteins in their relationship between aging and different traits. And we use this information to do an additional round of machine learning that's really focused on cognitive outcomes. So we apply this algorithm to another independent cohort where we're trying to look at how is the relationship between aging and cognition measured by these models. And then we use this information for feature selection to generate a second round of aging models, which we call cognition clocks. And then we can kind of go through the same procedure as I just walked you through on the previous slides in terms of evaluating these cognition clocks. So the first thing we did was just look for associations between the age gap on these clocks and Alzheimer's disease. And we have two Alzheimer's disease cohorts to look in. So we looked for clocks which have a robust association across these two cohorts with the age gap. And we have 4 clocks that meet this category. So this is the artery, brain, organismal clock, and the pancreas clock. And so the cognition brain clock has the strongest association with Alzheimer's disease across these two Alzheimer's cohorts, where again, people with Alzheimer's disease have about a half standard deviation increase in their cognition brain aging. And then again, looking now in sort of forward in time to predict how people will progress in dementia, which is something that the field is very interested in. We developed a multivariate model where we're trying to predict future dementia progression in Alzheimer's disease patients. And we found that our cognition brain age gap is a strong and independent predictor from the other known predictors that we have for Alzheimer's disease progression, notably plasma p-tau, which is sort of the current best research method that we have for predicting future Alzheimer's disease progression. And since it's actually somewhat independent from plasma p-tau, we can combine information from cognition, brain aging, and plasma p-tau to develop a new risk score for Alzheimer's disease progression, where if you have high levels of both of these things, then you're at, compared to average, you're at, again, about a 280% increased risk of future disease progression over a 5-year period. So we also wanted to take this a little bit further and really focus in on the early stage of cognitive decline. So we used some other information from some of our independent cohorts to look at cognition in healthy older people and the transition to dementia. So we again see now looking in another independent cohort, this is all people who are cognitively healthy and older. We can see there's a relationship between organismal, brain, artery, and pancreas aging in cognition. So people who have an accelerated organismal or brain age gap, for example, tend to have a lower cognition performance than older people who have a neutral or lower age gap. And then we can also look at specifically at this transition from healthy cognition to dementia. So we look again over a 15-year follow-up period in the NIGHT ADRC, which is one of our Alzheimer's disease cohorts. And we look specifically at people who entered the study as healthy controls, but over that 15-year period actually developed early Alzheimer's disease. And so when we look at which models actually predict the future conversion from healthy to early Alzheimer's disease, we see that in particular, models that are able to predict this are the artery clock and the organismal clock. And so we wanted to understand this a little bit further. And so I'm going to tell you a little bit of a vignette of diving into the mechanism of this. So we looked into the sequence of aging in these models by looking at how the proteins in these models change across the life course. And what we see is that the top model proteins are coming really from, again, from this organismal and artery aging clocks. And these are the ones that are predicting this dimension transition. And we thought this was interesting because we recognize some of these proteins from past research in the lab. It's important components of the blood-brain barrier. So using single-cell RNA sequencing data from brain vasculature, we saw these proteins were highly expressed in cell types that are specific to the blood-brain barrier, such as parasites, smooth mesocells, and the blood-brain barrier fibroblasts. So we know actually from previous work that these cells are specifically lost in Alzheimer's disease, and this leads to blood-brain barrier dysfunction. And this suggests a model where one of the early age-related changes in the brain that leads to dementia is changes in the cellular integrity of the blood-brain barrier that could be mediated by these proteins. So we also saw that early changes in these proteins in our models were enriched for matrix remodeling and vascular calcification functions. And indeed, most of these proteins have some known role in vascular calcification, which is considered a pathological process. So this suggests, again, also that extracellular matrix remodeling and vascular calcification are important events in brain aging that predispose people towards pathological outcomes. So just to summarize again, we can look into some of the underlying dynamics of the proteins in these models to develop new hypotheses for events that lead to the initiation of brain disease. So that's it for this portion of the talk. Thanks everybody for staying with me. I see that we're a little bit over time, but I appreciate you tuning in and coming here in person. So just to summarize this portion of the talk, hope we showed you that this is an exciting new approach for measuring organ aging using plasma proteomics, that we see many different kinds of organ aging types based on this method, that this method can be used to predict future disease risk and generate hypotheses for the underlying disease process. So just quickly in conclusion, I hope that I have convinced you with these stories of the power of the proteomics approach. Some of the key contributions of this work have been the development of new biomarkers and new methods. So we think this organ-based deconvolution of proteomics is something that could be applied in other disease settings as well. And we've seen some new biology through this approach, and you can look into some of our publications for more information there. So some of the near future work that I'm excited about here, one is to try to add more omics. So I think that we could probably do even better in understanding aging processes by looking across not just proteomics, but also these other methods that other groups have spent a lot of time working on. A major question, especially for the Alzheimer's and Parkinson's disease work, is what's the best way to combine multiple markers to get the best sort of diagnostic and prognostic testing? And then something that's sort of near and dear to my heart is now asking questions about the relationship between human genetic variation and organ aging. And I think another really exciting question that I hope the lab will dive into in the future is looking more at the temporal sequence of aging. So if we have data across the whole lifespan where we can sample people multiple times, can we see evidence of some organs aging earlier? And does that affect the aging of other organs in the future? And then finally, can we use this approach to try to identify drugs or interventions that can slow or even reverse the organ aging process? So with that, just some quick acknowledgements. Big shout out, especially to Hamilton O, who is a co-lead author on the organ aging study, as well as my mentors, Tony and Stephen, and Kathleen Poston, who's a neurologist here at Stanford, who was a major part of the Parkinson's disease work. And then thanks to some of our collaborator labs on the aging and Parkinson's disease studies, of course, everybody from the Montgomery lab, and then all the participants in the studies that I was analyzing today, as well as the leadership for those studies for putting the cohorts together. Very quickly, just some personal acknowledgements from the, the PhD journey side. Really thanks to all of my family who's here today, many of you in the audience. My mom, Mary, my dad, Shelby, my brothers, Dalton, Ryan, Rachel, Darius, and Delilah, my stepmom, Christy, and my stepdad, Jeff. I definitely couldn't have made it through this journey without all of you guys. And thanks for all of your support, especially during the pandemic and just all of the ups and downs that we've been through. There have been a number of friends who have really helped me along the way as well. So here's some of the major players. In particular, I just want to give a little shout out to Connor Horton, who I really appreciate. He has just been sort of a really important brainstorming partner and partner in crime through science for the last 5 years. And he's been such a huge help on days when I've been down and just helping me think through my projects. So huge thanks there. Also, there's been many other mentors who've helped me along this process. Particularly want to thank Alice and Posu, who were professors that I worked with originally when I came to Stanford, and their support through the beginning of my journey is, I think, definitely what made this possible. So I really appreciate their help as well. All right, and that's it. We made it. Thank you all for tuning in. I think we could take a couple of questions. And if someone could monitor the chat, if there ends up any questions in chat, but. Congratulations. Yeah. Thanks, guys. Nana is in the audience.
Very exciting. In the first part, especially encouraging when you can validate your kids in the cohort that you don't about treatment. I was wondering, would you consider to validate these kids in using ELISA or other orthogonal methods and be more easier for clinical usage?
Yeah, absolutely. I think that's a great question. So the question is, this biomarker DDC that we discovered in Parkinson's disease, have we considered validating with any other more targeted approaches that might be more applicable to the clinic, like ELISA? And I think that's definitely something that we're interested in doing. I think that's also probably important for really mapping out the full characteristics of the assay as a clinical tool or as a research tool, because it's possible that we'll just see actually better performance in those sorts of assays as opposed to the discovery-based proteomics that we've done. So I think that's definitely something that we're hoping to do in the future.
There are a couple questions online. So the first one's from Aditi Goyal. She's asking, can you elaborate on how you chose which machine learning models?
Yes. So the question, I guess for chat, I'm responding to Aditi's question on elaborating to machine learning models that we looked at in the aging study. So we were partially trying to prioritize accuracy and also just trying to prioritize interpretability. So we tested a number of different approaches, and we found in general that the model that we're using is a bagged lasso, which is basically a bootstrapped ensemble of lasso models, and lasso is a sparse linear regression model. And what we found is that these linear models, which maybe don't in theory capture as much of the sort of nonlinear change throughout the lifespan as a more expressive model like a neural network, what we found is that the sort of absolute performance gain that you get from these more complex models isn't that large. and you just have a lot more interpretability and sparseness with the linear models. So we decided to focus on that for a first pass, though I think it's definitely like an interesting thing to explore in the future, using more nonlinear methods that we can try to capture the true dynamics of specific proteins in plasma. Thanks, Emma.
I wonder if you could
Yeah, great. So the question is if I can speculate a little bit on the mechanism for why DDC is elevated in Parkinson's disease patients. And I think there's there's probably a number of different hypotheses there. I think my favorite is that I think is the most likely is that it's probably some sort of compensatory response to the loss of dopaminergic neurons in the midbrain. So it's possible that other places in the brain or other places in the periphery are trying to upregulate the production of this enzyme to try to respond to the dopamine deficit. It's also possible that some of it is actually reflecting active neurodegeneration of midbrain neurons. This is something that we see in markers like neurofilament and some tau as well. And so I think whether or not this is the case for PD in this marker is definitely an open question. And I think it's a little bit less likely because it's a pretty small population of neurons. But it is, I mean, the method that we're using is pretty sensitive. So I don't want to rule that out as a possibility. So. Rachel?
I don't know if you have access to data, but for when you look at the curve, you look at the average impact.
Yeah, that's a good question. So the question is, if we do look at the dopamine treatment, is there an effect essentially of dopamine treatment on the DDC levels? And we definitely do see one. So we see like, I think it's a hard thing to disentangle exactly how strong the effect is at this point, because we have some samples from the de novo cohort who aren't on medication, and then we have samples from people who are on medication. And we don't have a whole lot of, it's a little bit difficult to sort out the temporal dynamics and the addition of medication at the same time. But we definitely see some additional elevation of dopamine levels, but it doesn't appear to be a particularly quantitative elevation. So if we try to control for the dopamine therapy dose, then we still see a strong relationship between between motor symptoms and DDC levels. And we don't see a particularly like steep sort of slope relationship between dose and DDC levels.
Very great talk.
Thanks, Michael.
Another question on DDC. You mentioned that it's also elevated in Alzheimer's patients. Do you think they're also having dopaminergic loss, since you said that it's really specific to those attention neurons?
Yeah.
And do they have motor symptoms?
Yeah, so that's a great question. So Michael's asking if, since DDC does seem a little bit elevated in Alzheimer's patients as well, is there some, like, what's the reason there maybe? So to make the DDC story slightly more complicated, it is the essential enzyme as the last step of dopamine synthesis, but it's also an essential enzyme in serotonin synthesis. And so it's not just in dopaminergic neurons in the midbrain, it's also in some other aminergic neurons in the midbrain and other like serotonin producing populations. So I definitely think that, with AD being a much more widespread neurodegenerative condition, we are seeing some change in the neurochemistry in AD. And so you could see either a similar compensatory response or maybe, again, reflection of active neurodegeneration there. And AD does have some, AD does have some overlapping symptomology with Parkinson's, especially the non-motor symptoms. Like they both have a lot of problems with mood, anxiety, depression, irritability, things like that. And also the reality is like many patients actually have mixed pathology. And so we haven't been able to or tried to disentangle that at all. Like if you look at autopsy, the majority of people with neurodegenerative symptoms in life at autopsy, they have evidence of more than one pathology. So I think there's a question there. So.
Very nice talk, and I have a question about the relationship. I just want to learn more about the relationship between biological clock and disease. Do you think very, very high biological clock always to the direction? The reason I'm asking this is like, for example, if you have a color like super old, like and then do you think all their organs no biological age? And is there a possibility that someone have a very high biological age?
That's a great question. I think it's a pretty complex question that I can speculate on, but I think we definitely need more research, like specifically in those centenarian cohorts, to try to sort out. So the question is if accelerated biological aging on the aging clocks is always related to disease, or if there are cases, for example, centenarians, where they are very old, and maybe we don't know what their aging clock profile would look like. Maybe it would be very young, or maybe it wouldn't. Yeah, I think that's a great question, especially thinking within the context of brain disease and sort of resilience to Alzheimer's disease and things like that. We know a lot of centenarians actually do have a lot of pathology in their brain and some pretty active neurodegeneration. So it wouldn't surprise me if aging clocks are picking some of that up and actually still think that they look pretty old. But also I think it's like, since we don't understand fully the mechanisms of resilience, I think it's possible that that's not the case also. So I don't think I can speculate too much. What I can say, we do see occasionally in our data on the aging clock study, in the organ aging clock study, that very low biological age, so having an outlier young biological age, isn't always healthy. So we see more relationships sort of there's sort of like a linear range, like a couple standard deviation negative, as well as going pretty high positive, but people who have super outlier low negative ages, which there aren't that many, but they don't necessarily always look like even healthier than people who have sort of closer to average, but maybe low aging. So I think there's definitely more work to be done to understand really the dynamics there.
I know a common hypothesis within the biological community is the impact of inflammation. Yeah, and I was wondering if any evidence of inflammation, whether cytokine production, or impacting your ability aging clocks, or if any cells
Yeah, that's a great question. So the question is like, how, if at all, are we measuring inflammation in these clocks directly or indirectly? And I'll say, to the extent that we're measuring it with most of the organ aging clocks, we're measuring it indirectly, just sort of the criteria that we put forward to generate the models. Most of the like cytokines and things are not really hitting our criteria for tissue specificity, because there's infiltrating immune cells in most tissues, which are probably kind of contaminating the RNA-seq data that we used. So that's something that we thought about a little, is like going back and making more like sort of fine-grained immune focus clocks. And we haven't done a huge amount of that work, I will say. To the extent that the clocks measure inflammation directly, the liver clock is measuring a lot of innate immunity, because it produces a lot of the complement system. So there's a lot of complement in the liver clock, and we do see some pretty interesting relationships there with complement, but yeah.
Got one question online.
Cool.
This is from Sasha and Maria, and they say, to explain one more time to us non-biology scientists how the proteins we use to generate the ages of organs, and perhaps more importantly, what the uncertainties on those age measurements are.
Yeah.
If you want to, you can, I'll answer the first one with them later, but the second one on the uncertainties.
Yeah.
And your estimates might be.
Yeah, that's a great question. So responding to Sasha and Maria's question, I'll follow up with you on how we generate the organ aging clocks again. But to speak about the sort of the uncertainty in the measurement, I think that's a really good question. And that is mostly coming from, I think it kind of there's probably two sources to the uncertainty. One is technical measurement in the assays themselves. So like how accurately are we measuring protein levels? And for the SomaScan assay, I would say for the majority of proteins on the assay that we're using, the measurements are relatively accurate, especially when you compare to other proteomics platforms. So one way to measure this is just to look at sort of replicates sort of across different runs of the assay. And that coefficient of variation is pretty low for most proteins, around like 5%. Which is a bit lower than other technologies like mass spec, but so there's definitely some uncertainty there. And then another source of sort of biological uncertainty is just in the population sampling that we have. And I think that definitely there are questions about sort of the makeup of the cohort that we're using to kind of generate these statistical machine learning models. So I think we're doing super exceptional for like older Europeans at this point. And we have some data from other demographics, but not quite as much. And we don't have quite as much data from younger people as well, because most people who are in these sort of proteomic studies are either in a disease context or an aging context, and so they tend to be older. So I think probably the uncertainty is a little bit higher, but on those other demographics, but we haven't quantified it in like a rigorous way.
I think that's it. So let's congratulate Jarod again.
Thanks, everybody.
Not quite done yet. At this point, we transition to the closed session with any more questions. We'll ask everyone except the examiners. But as Tony pointed out.
Yeah,
maybe
thanks.
one of the- Yeah, so reception, building.
For those who are online and local, there's a reception at 11:30 at ChemH. If you want to stop by, 11:45. All right.
Good luck.
Thanks.